friends, welcome to the Point. In this session, we are discussing about applications of biotechnology. Biotechnology, it is concerned with the exploitation of biological components for production of useful products. Biotechnology, it refers to the development and utilization of biological process in the forms and systems that obtain maximum benefits to the man and other forms of life. Scientifically, biotechnology is defined as the controlled use of biological agents like microorganisms or cellular components for beneficial use. The Organization for Economic Cooperation and Development describes biotechnology as application of scientific and engineering principles to the processing of materials by biological agents to provide goods and services. An organism that passes on its characteristics to its offspring through basic units called genes. It is the pattern of these genes in plant or animal that determines the potential physical characteristics, growth and behavior of a particular organism. Biotechnology manipulates these genes in an organism to change its characteristics. For example, biotechnology can be utilized to produce new varieties of plants and animals with desired characteristics. Development of biotechnology took place in two phases: traditional biotechnology and modern biotechnology. Traditional biotechnology, it refers to conventional technology, that means which was being used for many centuries. It includes the process which is based on natural capabilities of microorganisms. Beer, wine, cheese and many such foods have been produced using traditional biotechnology. Modern biotechnology. This method is used for genetic modification of basic cell pattern in an organism. The utilization of genetic machinery of life for the production of special substances is called genetic engineering or biotechnology, or it is also called as gene technology. The genetic modification of microorganisms, it is so vital for their utilization in the production of useful biochemicals, and it can be brought by simple recombination or genetic manipulation. Uses of biotechnology. In agriculture, biotechnology is used for plant nutrition, pest control, protection of plants from climatic stress and disease, and in food industries, biotechnology is used for microorganisms to improve fermentation to produce enzymes from food industry and modified microorganisms for milk industry. In the field of healthcare, biotechnology is used for microorganisms to provide vaccines, antibiotics, etc. Protection of environment. Bioremediation of polluted environment, it reverses desertification of land and reverses greenhouse effect. By the use of this genetic engineering, today we produce a modified crops, new variety crops of plants, exotic vegetables, biofertilizers and biopesticides. Food preservation and flavoring are accomplished through the introduction of suitable microorganisms. Use of microorganisms. Microorganisms, they are used to produce solvents like ethanol and older alcohols, organic acids, carbohydrates, vitamins, etc. Microorganisms are used to produce desired changes in certain types of compounds or materials, and even in the production of electric power in fuel cells. Genetically altered microorganisms are used to produce insulin and interferon. Microorganisms are also used in the treatment of sewage sludge and in combating water pollution. Bacteria have been used for fixation of atmospheric nitrogen for agricultural benefit. Some of the products of biotechnology are mentioned below. Alcohols. Louis Pasteur demonstrated that fermentation of the sugar, it is carried out by microorganisms. Yeast ferments carbohydrates efficiently into alcohols. Microorganisms like bacteria and fungi, they are used to produce alcohol on a very large scale. Ethyl alcohol, it is produced from organic material and it contains sugar. The cost of the raw materials in fermentation, it affects the cost of products. Yeast are the main producers of ethanol, and they have been used as a major biological tool for the purpose of formation of ethanol. Fermentation process, it was discovered by Pasteur, and during 1980s this fermentation froth, it was discovered in sugar solution after the addition of yeast extracts, and this was the evidence for biochemical process of ethanol in the presence of yeast cells. Following are the uses of alcohol. Ethanol is used as a solvent, and it is used as a raw material to synthesize many other solvents like dyes, paracetamols, lubricants, detergents, pesticides, etc. Ethanol, it is used in alcoholic beverages such as wine, beer and rum. N-butanol, it is used in the manufacture of brake fluids and petrol additives. Glycerol, it is used in paracetamol and food industry. Alcohols are used as liquid fuel and it is called as gasohol. Biomass, it is derived from agriculture or municipal waste and it can be converted into alcohol for commercial use. Antibiotics. Antibiotics are chemical substances and it is secreted by living substances which kill or inhibit the activity of disease-causing microorganisms, for example penicillin and streptomycin, etc. The antibiotic penicillin, it was discovered by Alexander Fleming in 1929 and its commercial production began during 1940s. Antibiotics, they are produced by both fungi and bacteria, but over 50% of them are obtained by Streptomyces. It is a variety in soil bacteria, and they are widely used for curing human ailments caused by microorganisms. Enzymes. These are proteins and they produce biochemical reactions. Enzymes are prepared by microorganisms, animals or plants. Microbial enzymes have two advantages over the animal and plant enzyme. Firstly, they are economical and they can be produced on a large scale within a limited time. And secondly, they use technical advantage in producing enzymes using microorganisms. Like, they are capable of producing a wide variety of enzymes and they can grow in a wide range of environmental conditions, and also they have a genetic flexibility and they can be genetically manipulated to produce higher yield. Vitamins. All microorganisms, during their active growth, they face synthesis vitamins and other growth stimulating compounds. When the synthesis of these stimulating compounds they exceed their requirement, they are stored in cultures from where they can be recovered. Man takes advantages of such activities of microorganisms and collects vitamins from them. And some of the base material of vitamins produced by microorganisms are carotene, which contains vitamin A, riboflavin that contains vitamin B2, sarbose in vitamin C and B12. Vaccine. In its inactivated state, vaccine is a disease producing organism, and when the vaccine it is administered to an individual, it induces him to produce an antibody to combat the pathogen, that is disease producing organisms, and this vaccine protects the individual from the infection in future, and the administration of a vaccine in an individual is known as vaccination or immunization. The virus vaccine production using cell cultures, it follows some procedure. The cells that is used as a host, it is infected with the virus that is concerned and incubated. After an appropriate period of virus multiplication, the culture medium contains virus particles and they are collected and processed. And then the vaccines, they are stored at low temperature. A rigorous quality control is maintained throughout the entire operation. Indigenous hepatitis B vaccine. India's first genetically engineered vaccine, UNI, which is against hepatitis B, it was developed in 1997 by a Hyderabad-based laboratory, Shanta Biotechnics Private Limited, and the cost of this indigenous hepatitis vaccine is only 1/3 of the cost of the improved vaccine. Insulin. Insulin, it plays a major role in human life. Human insulin, it is essential for cell growth and it is used in treating osteoporosis, diabetes and acquired immunodeficiency syndrome (AIDS). The plant produced insulin, it is likely to be effective in curing other human diseases. Researchers have studied and established that transgenic plants, they could have been developed as useful tools for producing insulin for further medical use and where high amounts of recombinant human proteins are in demand, and these proteins, it can be produced either in greenhouse or field grown crops, and these studies show that transgenic rice and tea plants, they can be used for producing insulin commercially. Potential of biotechnology. Biotechnology, it is the exploitation of natural resources at the microbial and molecular level for the benefit of mankind, and it holds a promise in the future in the form of new drugs, therapies and bioremediation of waste. Human Genome Project. This project, it helped to determine the exact chemical sequence that constitutes the DNA in every cell of human body. Using this sequence, we can have the following advantage. Doctors, they could drip the droplets of our genes into a biochip to figure out if we have a cancer that could kill or a disease that would respond to a particular drug. Scientists, they could analyze selective genes from the parents and they could design babies even before their conception. Medicine. The role of biotechnology in the field of medicine is prevention of disease through vaccines, drugs and pharmaceuticals, delivery system and diagnosis, eradicating disease and creating new medical technologies. Some products like human insulin, human growth hormone, alpha interferon and hepatitis B vaccine, they are produced through genetic engineering. And another important product, it is known as tissue plasminogen activator, and it is being used to dissolve blood clots and thus to control heart attacks. It is found that genetically engineered mice cells, it is used for curing brain tumors, and gene therapy, it is another important treatment for certain diseases like thalassemia and for which no other treatment is available. Genetic diseases. Genetic diseases are those diseases or some abnormalities that is caused at before the birth by deformed genes. The result may be a defect in some organ or part of the body. Albinos, they are the result for the absence of pigment melanin when the color genes do not work properly. Hemophobia, it is caused when certain genes fail to produce blood clotting substances. Dwarfism, it is a result of abnormal growth of genes. Many of these diseases, they are incurable and patients generally have to live with abnormalities. However, with the help of biotechnology, there is a ray of hope that has been emerged in the society for treating some of these diseases. Tissue culture. Tissue culture is a technology of artificially growing cells, tissues and organs to the desired genetic purity and with that properties such as high yields and disease resistance. So it is important improving agriculture, crops and plants, forestries and for isolation of medical compounds used in pharmaceutical industry. Bioming, which is also known as microbial leaching. So and this is a process by which metals are being dissolved by microorganisms from ore-bearing rocks. Microbial technology, it recovers metals which cannot be economically processed. The low-grade ores, they are discarded in the waste heaps and that enter in the environment. The low-grade ores, they contain significant amount of nickel, lead and zinc and that could be processed by microbial leaching. Improving the quality of environment. The environmental biotechnology, it applies genetic engineering to improve the efficiency of waste treatment, and it reduces the environmental burden of toxic substances. Bioremediation. Bioremediation, it refers to the process of using microorganisms to degrade waste matter, and this can be done by introducing nutrients to stimulate the activity of bacteria in the waste, or by adding certain new bacteria to the soil. On this basis of removal and transportation of waste for treatment, there are two methods for degrading the waste: in situ bioremediation and ex situ bioremediation. In situ bioremediation means treatment of the waste in the place of its origin, and ex situ bioremediation means its treatment will be outside its place of origin. In situ bioremediation, it means conversion of environmental pollutants into harmless forms by introducing microorganisms. The ability of the surface bacteria to degrade a mixture of pollutants in groundwater, it is dependent on the concentration of compounds and duration of bacteria exposed to contamination. And sometimes it requires the supply of materials that stimulates the growth of microorganisms. Ex situ bioremediation, and this involves removal of waste materials from their place of origin and their collection at one place to facilitate microbial degradation. Solid waste treatment, and this includes waste such as leaves, animal and agriculture waste, and non-organic waste. The traditional cleanup practice involves informal processing of the organic materials by composting. Insects like earthworms, fungi, bacteria and other living organisms break down dead plant and animal matter and turn it back into the soil. Vermicomposting, it is the phenomena of compost formation by earthworms. And earthworms, they play a major role in the cycling of plant nutrients, turnover of organic matter and maintenance of the soil structure. They consume biomass. The most important effect of earthworms in agro ecosystem is the increase in nutrient cycling, particularly nitrogen. Decomposed organic matter, it is sustainable biofertilizer generated from organic waste. Sewage treatment, the contaminated solid matters, microorganisms and water is formulated into slurry. Thus, slurry phase treatment involves three major components: water, suspended particulate matter and air. In this process, water serves as suspending medium, where some contaminants are dissolved. Suspended particulate matter is heated as a solid waste. In a water treatment lagoon, air is provided for microbial growth, and the microorganisms break down the polluted organic matter, and sometimes more such organisms are introduced to enhance the biodegradation process. Hydrocarbons. Oils have variety of hydrocarbons, and if present in the environment, they cause pollution. Oil is insoluble in water and it floats on the surface of water. The microorganisms which are capable of degrading petroleum includes various bacteria and yeast. And there are two methods for bioremediation of hydrocarbons or oil spills. First, by using a mixture of bacteria, and second, using genetically engineered microbial stains. Toxic waste. A variety of toxic pollutants, they are being discharged to the environment from a large number of industries. For example, textile industry, it alone contributes a most of significant pollutants to the water bodies. Traces of zinc, cadmium, mercury, copper, chromium, lead are found in dyes. White rot fungi produces such enzymes and that degrade phenolic substances. And these enzymes, they convert and biotransform phenolic compounds in the wastewater to harmless compounds. White rot fungi, it also processes a variety of other substances that will degrade the pollutants efficiently. Improving human life. let's discuss how biotechnology improves human life. This biotechnology, it has lot of potential for improving human life in the future. Biotechnology, they are used to produce high yielding seeds by using gene therapy. Animal husbandry is likely to get benefited under a variety of complex methods like cloning, embryo transfer, etc. to produce specific breeds. Biotechnology helps in eradicating crop disease, making of plastic, paints and artificial fibers by using enzymes instead of chemical processes. Biotechnologists, they are developing new methods for the treatment of air pollution by industries, treatment of industrial effluents, microbial treatment of oil spills and toxic wastes. Biotechnology is a great asset in the field of health by producing genetically engineered vaccines. That's all for the session. Thank you.